Hello, everyone, and welcome to today's live broadcast, Assessing the Role of Prefrontal Cortex in Consciousness via a New Metric of Functional Connectivity, presented by Megan Peters, Ph.D., an Assistant Professor of Bioengineering at the University of California, Riverside. I am Judy O'Rourke of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Peters. I will now turn the presentation over to her.
Thank you very much for having me here at Labroots. I'm very excited to talk to you about the new metric of functional connectivity that we're developing here. Before I begin, I want to very quickly acknowledge and thank everybody who has made this project possible. I'm working closely with a number of people at the University of California, Los Angeles, as well as researchers at ATR, where the data were collected, and at Nagoya University. And so I also want to greatly thank all of the support and technical staff at ATR, that's the Advanced Telecommunications Research Institute International in Kyoto, Japan, and some other researchers who are not specifically involved but are really providing ongoing support and guidance. The data I'm going to be presenting today will also, or they were also collected with the support of the funding sources that you see at the bottom of your screen. And also a disclaimer, this is very much a work in progress, the data that I'll be presenting today. And so I encourage you to ask questions, provide feedback, and hopefully we'll get to some sort of interesting results later. So without any further ado, let's get started. The question that we wanted to answer with this project, which is really fundamental to the study of the neural correlates of consciousness, is how is it that the brain is able to bring a low-level visual representation into awareness? So what I mean by this is what computations or brain regions are responsible for the fact that we can become conscious of what our early sensory areas are representing. So there are a lot of theories about what makes something conscious. So we have, for example, global workspace theory, local recurrency theories, integrated information theory, and so on. But the one that I tend to subscribe to is what's called higher order thought theory. So in this theory, for something to rise into conscious awareness, prefrontal cortical regions must have what's called a higher order representation, which implies that they have access to the information in some way. So what I mean by this is when a lower order sensory area like visual cortex or ventral temporal cortex is representing some information like this dog here, that for you, the guy in the driver's seat, to become consciously aware of this thought, your prefrontal areas must have some sort of access and therefore form a representation of this dog as well. So this theory has a good deal of support, but one kind of obvious way to test it would be to see whether PFC having access to those low-level representations means those representations are consciously accessible. Okay, and we do have a good deal of evidence that this is potentially the right theory, although it is far from canon. Even though it's still controversial, we see that maybe the prefrontal cortex is in fact necessary for achieving conscious awareness. So some lesion data, so this is where the brain is damaged, this is in support of this theory. So, for example, you can see that bilateral lesions to the occipital cortex, so this is at the back of your brain, this can cause what's called cortical blindness. So you can't see, but you still have preserved awareness via other modalities. You're just as aware of your surroundings via touch, via smell, and so on, via hearing. So it's not really damaging your awareness, it's just damaging your vision. Likewise, you can see that bilateral parietal lesions, so this is kind of at the top back of your brain, this can cause what's called valence syndrome. So this syndrome is typically characterized by an inability to perceive multiple objects in the visual field simultaneously, but again, this leaves basic sensory awareness pretty much intact. You can also see that patients with bilateral orbitofrontal lesions, so this is at the front and the bottom of your brain, this can cause problems with social cognition and social regulation, but again, not sensory perception or consciousness. In contrast, though, patients who have bilateral prefrontal lesions or PFC lesions, they're less conscious in the sense that they're awake, but they're not in a coma, but they really lack the ability to engage meaningfully with the external world, with what might be thought of as a normal level of conscious awareness. So all of these lesion studies really do point to a pretty reasonable and meaningful role of prefrontal cortex in giving rise to conscious awareness. And some previous work that we actually did using electrocorticography also supported this view. So electrocorticography is where you record directly from the surface of the brain. So this is in the context of surgery for drug-resistant epilepsy. Neurosurgeons will actually remove the entire top of the head. You take off the top of your skull and you place electrodes all over the brain and you record neural activity while people are looking at stimuli or doing some task. And what we showed in this study is that when it comes to confidence judgments about perception, so these are kind of the metacognitive or awareness judgments about what you subjectively are seeing, that we see a much more distributed pattern in terms of which cortical areas are involved. So the bottom panel on the right of your screen shows that when you're judging confidence, all of your brain is really involved and that we see a distinct role of frontal regions. But when we're looking at just the perception itself, that you see a much stronger role of occipital cortex. So it seems that once again, we have some reasonable evidence that prefrontal cortex may be actually involved in conscious awareness. Okay, so this is the theory about prefrontal cortex's involvement in conscious awareness. But again, the way that we need to test this theory is by showing that when prefrontal cortex has access to low-level visual representations, that those representations then become conscious. The problem, however, is how do we define or measure what prefrontal cortex has access to? And then we need to also define this measure on a trial-by-trial basis, so that we can say, on this trial, you were consciously aware, and on this trial, you were not consciously aware. So that's really what we need to do here. So to answer the first part here, we can say, or we can define what prefrontal cortex has access to as what prefrontal cortex is functionally connected to. So functional connectivity, in its most basic form, in especially fMRI research, the traditional way to measure this functional connectivity between two regions of the brain with fMRI is to really just measure the covariation in the blood oxygen level dependent signal, or the BOLD signal, over time between these two regions. So you take the two regions and you measure the response of those regions to a particular stimulus, and you just correlate the response in time, and that gives you a measure of connectivity. And what we can do is we want to measure how that connectivity changes over time. So now this is called dynamic functional connectivity. And the principle is the same, only you can do these correlations now between two regions of interest, or ROIs, in a sliding window. So you pick a sliding window length, which is typically between 30 to 60 seconds on average. And you'll measure the correlations between two regions of interest within that time window. And there are other approaches, of course. You can do time frequency analysis, especially with imaging modalities that have higher temporal precision than you get in fMRI, where every picture is every 2 seconds, essentially. There are a number of reasons why this dynamic functional connectivity is really powerful. But for the purposes of our approach to measure what prefrontal cortex has access to on a trial-by-trial basis, there are some really kind of obvious limitations that we would need to address. The first one is that this 30 to 60 second time window, it really doesn't give us much temporal precision at all, especially if we want to measure the trial-by-trial connectivity, and our trials are less than a whole minute long. And additionally, the window size remains arbitrary. You know, the reasons to pick 30 or 60 seconds can kind of differ from one study to the next. And you can also decide whether you use a rectangular window or a window that kind of tapers off at the edges of time. So we wanted to see whether we could improve on these existing methods and to find a way to measure essentially near instantaneous functional connectivity between two regions using fMRI, or functional neuroimaging. Okay, so that's the necessary first step in our roadmap here. In order to measure whether consciousness depends on trial-by-trial connectivity between prefrontal regions and lower order perceptual areas, we have to define a metric that will give us the temporal precision we need using fMRI. We can then see whether this metric changes over time as a function of training, for example, and then we can ask our critical question about whether functional connectivity between PSD, or prefrontal cortex, and these lower order visual areas is actually related to conscious access. Okay, so this is our roadmap. So let's start by defining this new connectivity metric, okay? So to define the new connectivity metric, we're going to start with a basis of representational similarity analysis, or RSA. So representational similarity analysis essentially compares the multi-voxel pattern. So voxel is a three-dimensional pixel, a picture of your brain. So we'll compare the multi-voxel patterns produced by one stimulus to those produced by a different stimulus. So, for example, I show you a face, and then I measure the multi-voxel pattern of activity in, say, ventral temporal cortex, or VT. And then I show you a house, and I measure the pattern of bold activity in those same voxels. And then to compare these patterns, to get an idea of how similar they might be, I can essentially just correlate the patterns across space. So I can line up all the voxels into a one-dimensional array, from voxel 1 to voxel n within a particular ROI, or region of interest. And then so I line up all the voxels from one pattern, one stimulus, and then I line up the same voxels from a different stimulus, and then I just correlate the patterns across space. I can just use a Pearson correlation. And this will give me an R value, which varies between 0 to 1. And I can use this R value to say how similar or dissimilar these patterns are. And this is great. And I could even decide to do this at every TR, every time the fMRI takes a whole brain volume, a whole image of your brain. And I could look at how those correlations change from one TR to the next, or one trial to the next. So this could be super useful in comparing the patterns across multiple stimuli within a given region. And assuming the region can differentiate between the stimuli of different categories, the voxels will do so. So for example, in this ventral temporal cortex region, if I run this representational similarity analysis, I should see a higher degree of similarity between the two dog images, because they're from the same kind of image category, than between the dog and, say, the horse image. Okay, and so that's great. And I can also do the same thing within a different region of interest that I care about, let's say a higher order region like PSC, prefrontal cortex. So I should probably find kind of a similar result, that the dogs will be more similar to each other in terms of their representational pattern than they are to the horse. But what if I want to compare across regions? I can't just line up the voxels and do a correlation like I did within a single region because it's not the same voxels, right? So they're going to respond in a different way. Maybe I even have a different number of voxels in each area because they're different sizes. Ventral temporal cortex might be bigger than the prefrontal cortex region of interest, or it might be smaller than the region of interest that I care about. So basic representational similarity analysis really isn't going to cut it here. So to compare between two regions of interest that have different voxels or even different numbers of voxels, we'd have to find something to correlate that isn't spatially dependent. So what if we could correlate the strength of the representation across different concepts, but in an abstract sense rather than a spatial sense? So this is no longer which voxels are active and how much and correlate the voxels. It's more like which ideas are active or which concepts are active and how much. And then we can look at how those patterns are similar between two regions. The problem is we're going to need a lot more than 2 or 3 ideas to correlate or compare between the two areas. The brain can represent a whole lot more than dog or umbrella or horse, and that's it, right? And the other problem here is that running a correlation with only 3 data points isn't really going to get us much useful or meaningful information. So we're going to need a concept strength measure for a lot of different concepts that will allow us to correlate them or compare them across this entire kind of conceptual space in the 2 completely separate regions of interest in the brain. So the question then becomes, how do we get this concept strength measure, which you see on the y-axis of these little cartoon bar plots that I've shown? Typically, if we're using multivoxel pattern decoding, we could get a binary or possibly trinary output, wherein we use a support vector machine decoding or a logistic regression. And this y value, which we'd call concept strength here, this can then be replaced by the probability of a particular concept or a particular idea. So this is a probability value. It's between 0 and 1. And these are normalized now so that when you add up all of the concepts, the probability for all the concepts, you get 1 out of it. So essentially we get a probability distribution. And then we could use metrics that measure the similarity or difference between probability distributions to compare the informational content between the two areas. And we need to do this in a way that we don't get only 2 or 3 categories out of it. We need to get a lot of categories. So we need to run what's called a multinomial regression or multinomial classifier. So we get, say, 40 categories instead of just 3. But here's where we run into another bottleneck. To train even a binary or trinary decoder, it takes a lot of data. So to train a decoder that has like 40 categories, we really need a huge amount of sample data to build a stable, meaningful decoder that actually has some chance of decoding the true probability of different concepts in a particular area and not just noise. But we can get around that possibly by using a lot of people instead of just one person. So now we have a lot of people view the exact same sequence of images to build the decoder, and then we can functionally align the voxels in various regions of interest. And then we can pool all the data from many subjects and use that to train a decoder. So now I'm going to tell us how we functionally align my voxels to your voxels so that we can take all of the data from your brain and all of the data from my brain and put them together into like one super subject brain. And I can do this now from a lot of people. Okay. So to do the functional alignment, the first thing we have to do is make sure that everybody sees the exact same sequence of images, of events. And this allows us to map the trajectory of activity of one of my voxels onto one of your voxels across time within the same region of interest, right? So we're interested in categorical representations, that is, to what degree a particular region of interest represents dog or cat or horse or chair or umbrella at any given moment. So we'll present our participants with a sequence of images which come from these various categories, and then we'll measure the neural response, the bulb signal, in response to these images in ventral temporal cortex, and in prefrontal cortex while these people watch the images. And to make sure nobody falls asleep in the scanner, here we go, and that they're paying attention to the categories of these objects, we'll ask our participants to look at the images and then to press a button whenever they see the category of the image changing. So in this particular sample sequence, we see dog, 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 and then we see horse, horse, horse. And so at the point where the category switches from dog to horse, the participant in the fMRI scanner would press a button. Okay, and we had 38 people do this. Oh, sorry, we're measuring the activity in ventral temporal cortex and prefrontal cortex while people are doing this. And then we had 38 people do this. Each person sees 3,600 trials. So this is 90 exemplars, 90 different cats, 90 different dogs in each of 40 categories that we care about. Okay, and so now we can do that functional alignment. So we can pool all of the data from the 38 people in order to train our multivariate machine learning decoder. So this functional alignment is called hyperalignment. It uses essentially linear transformations, scaling, rotation, and translation to find the best mapping between the voxels in my brain and the voxels in your brain within a region of interest in terms of how they react to the stimuli. So it finds the voxel in my brain that responds this much to dog and this much to cat and intermediate to horse, and it finds the voxel in your brain that responds in a similar way, okay? And it does this using these linear translations here, or transformations here. Okay, then it's going to do the same thing, mapping the voxels of a third person onto the transformations that it found for you and me, and so on for all 38 people. And we'll do this for 1,000 voxels in each region of interest to find a common space for all participants. So these voxels now represent the voxel in my brain and in your brain and in that person's brain over there that all respond very similarly to all of these categories. So the beautiful thing about this now is that once you've trained these transformation matrices, you can actually transform the data from every single person into this common space. And now you have 3,600 images times 38 people, which is 136,800 trials. And this is actually maybe enough to train that 40-way classifier that we really care about. Okay, so the next thing that we have to do is we have to train the classifier and make sure that we have enough data to actually do that. Okay, so we could have used lots of different machine learning approaches, but we chose logistic regression because it has the advantage of simplicity. Its output literally represents the probability of one versus another category. So in binary logistic regression, what you get out of the fitted model is the probability of category A, and then probability of category B is 1 minus that. But in multinomial logistic regression, what you get out of the fitted model is a probability distribution. It's a distribution over the possible categories that the model is trying to predict. So essentially, this is the likelihood that the multivariate pattern that you're observing in ventral temporal cortex, VT, or PFC, which is prefrontal cortex, this is the probability that that pattern in that region of interest represents this particular conceptual category. So that's what I plotted here, again, with some cartoon bar plots. So you can see that ventral temporal cortex or prefrontal cortex, what you get out of the decoder is this probability distribution. So the ventral temporal cortex in the cartoon that I've created is probably representing snake and is less likely representing bee and is less likely representing giraffe, whereas prefrontal cortex is very strongly representing bee and very not strongly representing snake. Okay, and we can decide whether the decoder is correct if the tallest bar, the most probable category, for each one of the exemplar images in that decoder construction session is actually the image that the person was viewing when that brain pattern was created. So this gives us a 1 in 40 chance of being right by chance. And then we can see whether our decoder is actually doing better than chance in predicting what the person was looking at on every trial. And if the decoder is doing a good job, then we know that the decoder is actually picking up on something meaningful in these patterns. Okay, and so what we see here is that in ventral temporal cortex, so this is six-fold cross-validated for those of you who care about that stuff. In ventral temporal cortex, we see that the decoder is doing massively above chance. So we have almost a 1 in 5 chance of being correct in just randomly guessing, or in guessing whether or what the category is that the person was viewing on a trial-by-trial basis. In prefrontal cortex, we're doing a little bit worse. Prefrontal cortex is notoriously difficult to decode, but we are actually still getting above chance decoding. Again, chance is 1 out of 40, and we're a little bit above that. So this shows us that these decoders are actually picking up on something meaningful in these two areas, which is great. And now we can take the output of these decoders, which apparently is meaningful, and that output, again, is a probability distribution across the possible categories or concepts that these areas could be representing. So we're going to use standard ways of comparing probability distributions to measure the relationship between these two predicted outcomes. So we will define as our new functional connectivity metric something called functional information transmission, or FIT. And this is essentially taking the Kullback-Leibler divergence, which is the match between those two probability distributions in terms of their shape. And then we will also take into account the entropy of these distributions. And this is essentially how spiky the distributions are. So we want entropy and the Kullback-Leibler divergence to be low. We want the two distributions that are predicted in the two areas to be very similar and very spiky. We want them to be very strongly representing whatever it is that they're representing and not very strongly representing other things. If the probability distribution were flat across all of the possible categories, then the decoder seems to be just randomly guessing, and that's not what we want. So by this definition, smaller fit is better. We want Kullback-Leibler divergence and entropy to both be small. Okay, so let's take a look at how FIT does now in capturing whether the person is viewing an image consciously or viewing an image unconsciously. And the way that we do this unconscious viewing is by taking two images from the same category, but they're different images. And we look at the pattern of predicted responses in ventral temporal cortex and prefrontal cortex when both of the areas are simultaneously looking at the same image, like this, versus when they're looking at different images from the same category. Okay, so we'll call online viewing. This is when both areas are viewing the same trial. And offline comparison, we take the patterns from different trials, but the same category in the two regions of interest. And we can see that FIT is significantly smaller or better for cases where both regions of interest are actually simultaneously and consciously viewing the same object. So this is our first indication that FIT actually may index something meaningful about conscious access or conscious functional connectivity between prefrontal cortex and ventral temporal cortex. So that's nice. And then we can also do a similar thing for comparing two different exemplars within the same category to two different exemplars that come from different categories. So if what this metric is measuring is the representational similarity in information space between these two regions of interest, we should see cases where the two dog patterns are more similar to each other than a dog and an umbrella or a dog and a horse. So this is very similar to the representational similarity that we talked about earlier. And indeed, this is exactly what we see. So we see that between category functional information transmission is always worse than within category fit. And we can confirm this with ANOVA and t-tests and other statistical tests like that. Okay, so this is great. We've now developed at least preliminarily validated a metric that may allow us to measure this instantaneous functional connectivity or informational similarity between two regions of interest. And we've shown that between prefrontal cortex and ventral temporal cortex, this metric does seem to index some things we care about having to do with conscious access to low-level visual representations. When two regions contain similar informational content, the FIT metric is able to capture that. And it also is able to capture whether the two areas are looking at the same thing versus different things from the same category. So this is really nice. Now we're going to be able to move on to 2 and 3 within our roadmap. And we actually designed an experiment that would try to provide answers to both 2 and 3 in the same paradigm. So I'm going to introduce that design now, and then I'll show you some results that we have from our pilot subjects. And again, as a disclaimer, this really is a work in progress. So the results that I just showed you, we have a lot of people, and we feel pretty confident in the results. But the new results that I'm going to show you, we only have a few people. So take what I'm showing you with a grain of salt. Just because we haven't, maybe come up with the complete answer yet doesn't mean we're not on the right track. Okay. So our behavioral paradigm, it's a real-time functional neuroimaging study. And in this study, subjects will undergo blocks of trials that look basically like this. So on every trial, we're going to first show you a dot, and you're supposed to look at the dot. This is while you're lying in the scanner. And then during what we're going to call the induction period, we ask participants to concentrate on changing their brain activity. And the goal of changing your brain activity during this period is to maximize the feedback that we're going to give you, which will earn you money. So during this, this induction period, we will decode the 2 regions of interest, the ventral temporal cortex and the prefrontal cortex, and then we'll calculate the fit between them. And then we'll show you a dot again, and then we'll show you some feedback. And this feedback, the circle size, is related to the fit that we calculated during that induction period. So if you did a good job, then you see a big circle and you get a lot of money. And if you did a bad job, you see a small circle and you don't earn very much money. And importantly, the circle size here, this is related to the functional information transmission. So if the fit is good between the 2 regions, if they're representing the same kind of thing, even though there's nothing on the screen, then we'll give you a big feedback circle. Okay. And the way that this works then is that we'll have, on one day, we'll do the decoder construction, and then we'll do 3 days of this decoded neurofeedback, or DECNEF. And I'll get to those probe trials that you see in the title in a second, but first we show people, 8 to 10 runs of 16 trials per run every day. So this is pretty intense and pretty exhausting for our participants. Okay. So we first wanted to see whether this decoded neurofeedback leads to better fit over time. That is, whether people can learn to better monitor their own low-level perceptual representations. So now, while we had 8 people go through the whole experiment, only 4 of them received multiple days of training in which the feedback measure was based on fit. We were piloting, so several subjects received feedback that was based on a slightly different metric. So we'll remove those subjects from our analysis, and now we have only 4 left, unfortunately. But now we're going to look at whether fit changes over time as a result of training. So with these 4 subjects, we don't see a whole lot of trends emerging in fit. However, there may be other trends in the learning that we can't see just by looking at fit. So for that, we're going to have to dig deeper into what it means to have conscious access to a low-level representation in the first place. Okay, so now we're going to move on to number 3 in our roadmap, and then we're going to go back and see whether any of those metrics change. So to answer whether people are learning to improve conscious access over the course of training, we have to actually define how we're going to measure that conscious access beyond just the fit metric itself. So now we have 8 people that we can look at for this, because the feedback for all of them doesn't matter for this measure of conscious access, which I'll show you right now. Okay, so in this real-time fMRI paradigm, we show the structure that I showed you before, but then we also have these additional probe trials that I mentioned earlier. So occasionally, instead of showing the feedback circle right away, we'll actually show 2 pictures on the screen. So these pictures are from the same categories as the decoder construction images that we saw earlier in the presentation. And we'll choose these images based on what the brain happens to be representing during this induction period. So during that induction period, that noise screen, we're decoding the 2 regions of interest. And then I'm going to choose the images that I'll show you in the probe trials based on the decoded probabilities of those categories during that induction period. And I'll get to how I actually choose or which images I choose in a second, but I want to point out that during this noise period, that induction period, where you're changing your brain activity, I'm not actually showing anything on the screen here, okay? But I'm going to lie to my participants, and I'm going to tell them that sometimes inside the noise, there is also a picture being shown. So this gets them to try to look for pictures within that noise, okay? And after they choose which image they think was embedded in the noise, they have to rate their confidence in their choice on a scale of 1 to 4. Okay, and then they get their feedback. And the feedback, of course, is unrelated to this choice and confidence kind of questioning. It's really just based on how well you were able to change your brain activity to match the conceptual representations between prefrontal cortex and ventral temporal cortex. And because we wanted to really focus on having people change their brain activity, we have only up to 30% of these probe trials on every given run on every day. Okay, so I told you I would tell you how these probe trial images are shown and chosen. So remember, we're not actually showing anything in the noise, even though we're telling our participants that we are. So instead, we're choosing our targets, so this is the correct answers, and the distractors, the wrong answers, on the basis of the decoding that we do during that induction period. So the target is always the most likely category from the ventral temporal decoding. So that is always what your ventral temporal cortex is most likely representing. So in the cartoon bar graph that I've shown here, ventral temporal cortex is most strongly representing snake. So snake would become the target on this probe trial. Now, the distractors can fall into 2 categories. On half of the trials, we choose the least likely category from ventral temporal decoding. So in this cartoon bar graph that I've created, this becomes the shark category. In the other half of the probe trials, we're going to choose the most likely category from the prefrontal cortex decoding. So in this cartoon bar graph, this becomes the B category. Okay, so we show all of these probe trials with essentially 50-50 probability, and then we measure choices and confidence in response. Okay, and then we're going to have 1 day of decoder construction, as we described earlier, then we'll have up to 3 days of that real-time behavioral procedure. Okay, so to look at whether prefrontal cortex connectivity predicts conscious access, we're going to use what's called choice probability analysis. So this type of analysis allows us to see to what degree a particular metric, in this case the fit metric, is diagnostic of an observer's choices. So can we predict what the observer is going to choose, what the person is going to choose, and other aspects of their behavior, like their confidence, using this particular metric? Okay, so our big question, of course, is whether fit, the relational similarity between prefrontal cortex and ventral temporal cortex, does this predict what people are going to choose? And so what we'll do to do this is we'll say, okay, if fit is very large, so it's very bad, we'll predict at this particular criterion that you're going to be incorrect. And then if fit is very good, then like it's a 2, then we'll predict that you'll get the choice correct between those 2 pictures. And so we can then plot what's called the false alarm rate against the hit rate, and we can call this particular criterion level at which we make these predictions A. And then we'll move that criterion and change our predictions. So now we predict that at an intermediate level of fit, we predict that you're going to be incorrect. And so this becomes a correct rejection, whereas for the point A, it was a false alarm. And so now we get a different dot on this curve, and we can say that the area under this receiver operating characteristic curve, so this is signal detection theory for you nerds out there, this is called the choice probability. Okay, so choice probability at 0.5 or 50% is purely guessing. Choice probability of 1 is perfect prediction, and we really want it to be somewhere in between. Okay, so now kind of the crux of the argument here. We're getting towards the end. So the first trial type we're going to look at is these against lowest ventral temporal cortex or against lowest VT probe trials. And the question is, does better fit mean that the observer is more likely to choose the target? And unfortunately, the answer does seem to be no. We did not see that choice probability is any different from 0.5. But again, remember, we only have 8 people going through this, and some of them don't even have 3 days of training, so we do still have a pretty limited amount of data. We did some other choice probability analyses to see if there were some other patterns that might be interesting in the data. So we looked at the probability of the target, so that's the height of that bar in the ventral temporal cortex, minus the probability of the distractor, so the height of that really super tiny bar in the ventral temporal cortex. And we don't see any patterns here either. And we also don't see that your confidence ratings meaningfully predict whether you're going to be correct or incorrect on a trial-by-trial basis. Now, in the against the highest PFC probe trials, maybe we have a slightly better shot. So unfortunately, we don't see any relationships between fit and your trial-by-trial ability to choose the target. However, we do now see that the difference between the height of the bar in ventral temporal cortex that defines the target and the height of the bar in prefrontal cortex that defines the distractor, the difference between these bars now actually does predict which one people are going to choose. So if ventral temporal cortex is more strongly representing whatever it's representing, then you choose the ventral temporal cortex target. And likewise, if prefrontal cortex is more strongly representing whatever it happens to be representing, then that's the one that you choose in this choice. We also see that there might be a little bit of predictive power in the confidence ratings, so that when people indicate higher confidence, they're more likely to choose that target. And now we can go back and we can use this choice probability analysis in subjects for whom the feedback was consistent and meaningful. And we can take a look at how their behavior in those probe trials evolves over time. So first, in these 4 people, remember we only have 4 people here, now we are starting to see that confidence ratings may be predictive of your choice even in those against lowest VT trials, and that we retain the ability of the difference between the height of the bar for the target and the height of the bar for the distractor. This still predicts your choice in those against highest PFC trials. We also see maybe that there is some learning in your ability to access this low-level representation over time. We just might not have seen it in the fit metric just yet. So we finally looked at whether the overall trial number, this is basically how far you've gotten through the training protocol, is this likely to predict your accuracy on this 2 alternative force choice? And the answer is yes, barely. So remember, we only have 4 people here, but we're starting to see that maybe with training, people do have the ability to gain access to their low-level representations. So now we need to see why our fit metric may not capture this and how we can refine our fit metric to be able to predict these types of behavioral responses. And as, of course, a disclaimer, this is a work in progress. We have a very small number of subjects, and so we want to temper our excitement about this until we have some further proof that it might be correct. Okay, so as a summary of what I've presented today, we have defined a new connectivity metric. This is done, we've validated it, although we do need to do some more work to refine it. We've targeted it with decoded neurofeedback to see whether people can learn to improve it, and we've tried to relate it to conscious awareness, and these are works that are definitely still in progress. So there are some open questions, of course, and future directions that our research is going to take. So we're going to further validate our fit metric. We're going to compare it to representational similarity analysis within each region of interest using, for example, k-means clustering. We're going to compare fit to traditional dynamic functional connectivity. We're going to relate fit to the default mode network activity. And of course, we need to collect some more data. So with that, I'd like to thank all of you for your attention. I'd like to once again thank all of my collaborators, without whom this work would definitely not be possible. And I'd like to thank Labroots for hosting this event.
Thank you, Dr. Peters, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for, so let's get started. Our first question is, have you ever thought about using hyperalignment to align VT and PFC to make fit work better?
Yeah, this is a great question that I've gotten before. So I think this is in principle a great idea, that we can look at how we can better align the representational content in VT and in PSC if we do some sort of hyperalignment to line everything up to begin with. The challenge here is that the way that ventral temporal cortex and prefrontal cortex represent these kinds of ideas might kind of cluster differently. And what I mean by that is that when ventral temporal cortex thinks about something that's kind of similar to a dog, it might care about things that have 4 legs. And so it might say dogs and chairs and tables are very similar to each other, and dogs and chickens and snakes are not very similar to each other. And in contrast, when prefrontal cortex, these are just examples, by the way, but prefrontal cortex might cluster concepts kind of differently. So maybe prefrontal cortex cares more about animacy than ventral temporal cortex does. So prefrontal cortex is going to say that dogs and tables are very dissimilar, even though they both have 4 legs, and that dogs and chickens and snakes are more similar to each other. So the hyperalignment is a linear transformation, and so there may be challenges in trying to deal with the linearity of that transformation that would not take into account these differences in essentially the barycenters of the clusters. So well, this is a great idea in theory, and we're looking into ways of doing this type of transformation to modify the fit and metric and make it better. We don't have the answer quite yet, but great question.
Next one is, what were some of the other versions of fit you tried, and do they work in predicting conscious awareness any differently?
Yeah, this is also a great question. The first thing that we tried actually was we didn't look at entropy at all. So entropy is essentially the spikiness of the probability distribution. So whether the probability distribution over those conceptual categories, it's very strongly horse, for example, and like nothing else, or whether it's just kind of like sort of horse and sort of chicken and sort of giraffe. And so initially we didn't use entropy, and we just used the Kullback-Leibler divergence, which is the degree to which 2 probability distributions are similar, regardless of their spikiness. And we found that one of my participants in particular was very smart in figuring out this task, and so she figured out how to make her brain represent essentially nothingness, all the categories at once. And so then the decoding showed that the 2 areas that we care about had very similar representations because they were very similarly representing nothing. And so we had to change the metric to incorporate entropy, or the spikiness of the distributions, in order to counteract that. And so we're looking at other ways of defining fit too. We tried just a straight Pearson correlation, and that didn't work really well. So we're still open to suggestions. So if you have any suggestions, please feel free to email me and let me know.
One more question. Could you use this metric in other neuroimaging modalities?
Yeah, this is also a very good question. In principle, you could. Any neuroimaging modality that would allow you to decode in a multivariate way and in a multinomial way. So you need to, the critical thing is that you need to have many different categories that you're decoding among. You can't just have a binary or a trinary decoder. But really, we're not tied to the fMRI imaging modality. So any imaging modality that would allow you to get kind of a multidimensional probability distribution out of it would lend itself to fit, or wherever fit ends up after we do more research on it. So yeah, you could use this with EEG. You could use it with a lot of other neuroimaging modalities in humans and in non-human animals as well, if you wanted to.
Again, I would like to once again thank Dr. Peters for her presentation. I would also like to thank Labroots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June 15th, 2018. You'll receive an email from Labroots letting you know when this webcast will be available for replay. Please share this announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.